Now that we know the basics of nucleic acid structure, let's go deeper and look at how that structure influences its function. We'll talk about things like base modifications. We'll talk about things like mutations. We'll talk about things like how even unmutated DNA is going to be wound up really tightly around these proteins called histones to form nucleosomes. And then if you want to actually access the DNA, you're going to have to move things away. And then we'll see how the structure of DNA, that double helix, is going to offer kind of view windows for proteins called transcription factors to come and help figure out when to use what. And so we'll talk about these things, but first we need to kind of talk about the functions. So let's talk about DNA and RNA. We'll talk about transcription and translation, just a broad overview. Make sure we're on the same page with some of the terminology. And then we'll go deeper and look at the actual biochemistry that's making these processes possible. Thinking back to the central dogma of molecular biology, we have a DNA gene that has instructions for making a protein. In order to actually get the protein making instructions to the protein makers, that gene has to first be transcribed into RNA. This is a process called transcription, and it takes place in the nucleus where the gene is. And then that RNA gets processed and becomes mature messenger RNA or mRNA. And then that mRNA goes out into the cytoplasm where the ribosomes, the protein-making complexes are, and the ribosomes are then going to travel along the mRNA and make a protein. Your mRNAs are only going to be single-stranded, but your genes, the DNA, is going to be double-stranded. One of those strands is going to have information that kind of makes sense for making a protein. We call that the sense strand or the coding strand. And then the other strand is kind of just the other half that's needed to make copies of that coding strand, of that sense strand, but it would be gibberish to the ribosomes. We call that the antisense strand or the template strand because it serves as a template for making the coding strand. So you have a gene that gets transcribed to make a messenger RNA, or I guess after processing, it makes a messenger RNA, and then that gets translated to make a protein. That's in the simplest case. There are some genes that get transcribed to RNA that acts as functional RNA, so it just functions as RNA. But in the case where you want to make a protein, then that RNA is going to be a messenger RNA, and it's going to basically get processed to form a mature messenger RNA. It gets a cap and a tail and some splicing, and then it gets to be used to make a protein. When it gets used to make a protein, how it works is that the ribosome is going to travel along the messenger RNA and it's going to read kind of 3 letters at a time. We call these 3-letter combos codons. So in your mRNA, every 3 RNA letters is going to be a codon. And so you'll have one codon code per amino acid. So if you look at a codon table, these are basically the 3-letter combinations, the codons that are then recognized by the tRNAs. So the transfer RNAs are one of those types of functional RNAs. And what they do is they actually they have a matching complementary word that's going to have like allow for base pairing with that codon. So we call this the anticodon. And then on their other hand, you can think of that as kind of like being their foot is going to have that anticodon that matches the sequence. And then their arm is holding onto an amino acid that matches that codon, that codon codes for. And it's going to bring that amino acid to the ribosome. And then that amino acid is going to get linked on to the growing chain in this process called translation. So the ribosome is going to move along the mRNA, or the mRNA is going to move through the ribosome, however you want to think about it. But what's going to happen is that it's going to keep opening up a site for a new tRNA to bring in a new amino acid. And that tRNA is going to have an anticodon that matches the codon in the newly exposed site. And it has the amino acid that matches that codon and the anticodon. And you'll see that some of the amino acids have multiple codons that code for them. Because if you think, okay, we've got 4 RNA letters and we have 3-letter combos, that's 64 potential combos. Some of those are stop signs, but most of them are coding for amino acids. That'd be a lot of tRNAs. And so let's consolidate. Let's have multiple codons spell for the same amino acid. And then let's be lazy and let's have the same tRNA be able to recognize multiple of those. So some amino acids will have multiple tRNAs that bring amino acids for them, and some recognize one codon and some recognize other codon. But there are also tRNAs that can recognize multiple codons, but always bring the same amino acid. And the reason why they can do this is because of this like wobble position. This third position is a little more kind of loosey-goosey. And you're often going to see those GU base pairs there, those kind of weaker base pairs that the RNA can form. And remember, DNA can't form those because DNA doesn't have U. But you're often going to see that GU in that third position, that wobble position, when we think about the tRNA, the codon-anticodon matching. So that's one of the ways in which RNA can be really great for allowing you to have more kind of flexibility. However, you wouldn't want that flexibility in your DNA because then you could get things like mutations. Speaking of which, the reason why I'm sometimes I'm trying to be careful about saying mutated proteins is because some people get mad because mutations happen at the level of the DNA, and why that distinction can matter is because sometimes those mutations have an effect on the protein that gets made and sometimes they don't. If you think about these three-letter combos, and we had multiple combos that can kind of spell for or encode a single amino acid, we call it degeneracy. So the code is degenerate, but it's not ambiguous. So each codon will only ever spell one thing, and it's not overlapping. So you basically go 3, 3, 3, 3, but then those threes aren't overlapping. AUG also, in addition to methionine, it also tells the ribosome to start here. And then if it's in the middle of a protein, it'll before you reach a stop sign, it'll just put a methionine in there. But if there's signaling stuff upstream that kind of tells it that that's a start, and then you have this AUG, it'll start there. And it'll keep going until it hits a stop. Sometimes what can happen is a mutation can introduce a premature stop, and this is what we call a nonsense mutation. There are also mutations that can change what amino acid gets encoded. We call those a missense mutation. And then there are mutations that don't change the amino acid that gets brought in because of that degeneracy. So for example, we can go from an AAG to an AAA, and we would still be encoding for a lysine. But if we go from an AAG to an AAU, well, now we're encoding a leucine. That's a missense mutation. And if we go from an AAG to an UAG, then we're encoding a stop sign, and that would be an example of a nonsense mutation. Those were all kind of point mutations where we're just swapping one position. But then you can also have indels, insertion and deletions. Because the codons are 3 letters, if you insert or remove multiples of 3, you're going to either gain or lose an amino acid, but you're going to stay in frame. So you don't get out of step. All the other things before and after it stay the same. But if you lose or gain something that's not a 3, so a 2 or 1, well, now you get what was called a frame shift mutation, where everything after it is kind of out of whack. There are various software tools and websites that you can use to see what the complements and reverse complements and the RNA sequences and the proteins that will get made are. And so Xpasy has various tools for this, as well as Benchling. There are a lot of different options you can use, lots of online tools. So if you want to do translation, Xpasy is really good. There's also this sequence manipulation site that'll do all sorts of manipulations for you, because you don't want to get confused when you're thinking about 5' to 3' on one strand versus the other strand, and your mind can get all mixed up. But these tools can be really helpful. But you lose a lot of the picture if you're just looking at the sequence, because the DNA structure goes way beyond the sequence. So let's now look and see how DNA wraps around proteins called histones to form nucleosomes, giving you kind of beads on a string. And then those wind up even further to give you really condensed chromosomes that can actually fit in your cells, as opposed to just a bowl of spaghetti that would overflow your cells. These histone proteins, basically they're an octamer, so you have 8 chains in this multimer. And they form this kind of little ball, you can think of it as. A ball of yarn with a bunch of ends sticking out, these tails. And these tails have a lot of lysines that are going to allow it to be modified, as we'll see. The DNA is going to wrap around this octamer, and we get this sort of thing that looks like a bead, and we end up with this beads on a string analogy, where we have this DNA wrapped up around these histone octamers, and all of this is going to be coiled up even tighter, the form of our chromatin, this tightly wound DNA that's going to be stored in our nucleus. When we talk about the DNA wrapped around a histone, we call this a nucleosome. And so we have lots of nucleosomes, and the histone proteins in those nucleosomes can be modified in order to alter the accessibility of regions of DNA. If we go back and think about our base pairing and look at what would be seen if we were to look into one of those grooves. So if we were to look at the major groove, what would we see? And if we were to look at the minor groove, what would we see? If we go and we look at what hydrogen bond acceptors and what hydrogen bond donors are kind of visible in each of those grooves, we'll see different things. And the things that we see are going to be dependent on what the sequence of the nucleotides are. So we're going to have our base pairs formed here, and then we are looking in at them from either side. From the major groove, if you were to look at a GC versus a CG, so one strand, say you have your left strand has a G and your right strand has a C, versus your left strand having a C and your right strand having a G. In both cases, you're going to have a G and a C, they're just in a different order. This different order is going to make it so that on the major groove view, you're going to see different things. So if you were to look at the major groove of GC, you would see AADH, where you have an acceptor, a donor, and a hydrogen. And if you were to look at the CG, you would see HDAA, hydrogen, donor, acceptor. But if you were to look at the minor groove, well, here they would actually look the same in both cases. You'd see an acceptor, a donor, and an acceptor. Something similar happens in the case of our AT pairs. From the major groove view, you'd be able to tell them apart, but from the minor groove view, you couldn't. In the minor groove, you'd still be able to tell apart a GC or a CG from an AT or a TA, so you can tell this apart from this, but you can't tell this apart from this or this apart from this. Bottom line, you get more information from that major groove, and it's also easier to access. And in fact, there are proteins called transcription factors that go and they look at that major groove, they bind to that major groove in order to regulate gene expression. Transcription factors bind in front of the gene and tell RNA polymerase, hey, come here, start copying this into RNA so we can make protein from it. These transcription factors kind of know where to bind because they're able to look in the major groove and find sequences that make them happy. The major groove is going to give them more information, and so they're typically going to form those sequence-specific interactions in this major groove. You'll see things like zinc fingers that actually go and they kind of interrogate or go in this major groove and form specific bonds to the base pairs. And we'll look in a second at kind of how they can do this. But then you also see interactions in the minor groove that help stabilize things. So here's another example, that transcription factor binding to this strip of DNA. So you can see that you're forming these more interactions in the major groove. I should also mention that sometimes these interactions are going to be mediated by water. So the DNA backbone, it's going to have a lot of water around it, as well as there'll be metals and things. But this water around the backbone itself is kind of going to be more ordered in like a solvent layer. And when you have, say, a protein bind, it can either bind by kind of recognizing that solvent layer of water, or it can also, if it pushes out that water, well, now you get an entropy increase. So this could also contribute to the binding affinity. We talked about how transcription factors can look in the major groove and make bonds there and recruit RNA polymerase to come and transcribe a gene. But in order to be able to see that major groove can't be tied up in one of these nucleosomes. So instead, these nucleosomes have to be able to be remodeled. There are proteins that can help with the remodeling, and there are also going to be changes we can make directly to the histone proteins to alter how tightly the DNA wants to bind to them. Thinking about lysine, remember we have that positive charge, and we said that the backbone of DNA is negatively charged. So these can form these ionic bonds with one another. Great. That's going to keep our DNA tightly wound up. What if we get rid of that positive charge? We modify lysine in a way that gets rid of that positive charge. We can do that by acetylating it. So by adding this acetyl group, this is going to neutralize the lysine. And if it's neutral, well, now it doesn't have that strong drive to hang out with the DNA. And so this can kind of loosen up our nucleosome so that transcription factors can gain access. But that's not all we can do to that lysine. We can also modify it in other ways, including methylation. In the case of methylation, here we're not changing the charge, but we are changing binding opportunities. We can make methyl marks then on these histones that might not change, like directly change how tightly that DNA is coiled, but it can offer opportunities for things like various chromatin remodelers to come bind and push away the chromatin or tie it up tightly. So you can have all of these interactions, all of this sort of integrated signaling that's coming from modifying those histone proteins that the DNA is wound around. In general, acetylation, because you're kind of axing the charge, you're making it so that the region of DNA is typically opened up more, so this can increase the transcription. And in the case of methylation, this typically gives you decreased transcription, but it's also going to depend on where the methylation occurs, both within those histone proteins. There's lots of different lysines, as well as what else is around it and where it's located and all sorts of complicated things, but that's a big area of research is on this histone modifications. When we talk about histone modifications, we're talking about modifying the lysines in the histone proteins that the DNA is wound around. And this is one form of epigenetic modification, genetic information that's on top of the original sequence of DNA. There's also a form of epigenetic regulation in which we're actually modifying the DNA itself. The nitrogenous bases can undergo modifications, both ones that you want and ones that you don't want. And so in terms of wanting them, often RNA is going to have a variety of different modifications that are going to help allow it to form different sorts of structures and allow your immune system to kind of recognize what's foreign or what's not foreign. In the case of your DNA, you can have methylation of the bases that influences how they get transcribed, in addition to having changes like methylation of the histone proteins that are wrapped around. In terms of the base methylation, we can have intentional methylation where our cells are using specific enzymes to place methyl groups at specific positions to have specific functions. And most typically, this is going to happen like a methyl cytosine. This can have important epigenetic effects on dictating sorts of what regions are going to be transcribed when, as well as you can actually get some inheritance of some epigenetic things. That's all complicated and I'm not going to get into. But basically, you can have the bases be methylated. And that would be a form of intentional modification. However, we can also get chemically induced methylation. Alkylating agents can cause this, so things like nitrogen mustard can cause alkylation, dimethyl nitrosamine, dimethyl sulfate, various compounds that can add methyl groups when you don't want them. And why might you not want them? Well, if you make it so that you're adding a methyl group, now you might confuse the transcriptional machinery or the copying machinery, the replication machinery that's actually making permanent copies of your DNA. Remember the DNA copy, that's like our permanent thing. That's the one we want to protect. And so if we have a modification that makes it so that the replication machinery, the DNA polymerase, when it's coming to make copies, if it doesn't know, it misrecognizes the base, it'll put in the wrong base on the other side, and this will lead to a mutation in the DNA that gets made, and then the DNA that gets made from that, and the DNA that gets made from that, and the DNA that gets made from that. If you have a modification that makes it so that like a mess up when you're doing transcription, that's not so bad. Or if you have a mess up when you're translating, that's not so bad because the RNA is going to be temporary. That protein is going to be temporary, but that DNA is going to be permanent. So we need to protect the DNA more than we need to protect the RNA. And we need to have really good proofreaders to make sure that we can fix those changes before we actually go and replicate it. Especially because it turns out that modifications happen a lot. So we talked about methylation where you're adding something, but another common modification, a really common modification, is when you're taking something away. And often what you're going to be taking away is that amino group. So you get spontaneous deamination, so loss of an amine group. And we're going to lose this extra cyclical one. So the one that's not part of the ring, but that's on the outside. If we think about our amine groups, we've got them on cytosine, and we've got them on adenine, and we've got them on guanine. The most common one we see is this deamination of cytosine, which happens about 100 times per day. So not per cytosine, but like in your body, this will happen about 100 times per day. Yet our cells are able to fix that. We have these mutations happening all the time, but our cells can fix them before they get kind of incorporated into our permanent DNA. And one of the ways that we know is because things will look out of place. So if you imagine that you deaminated cytosine, what you'd end up with was actually uracil. And so your DNA copiers know that uracil does not belong in DNA, and so they can go in and fix it. You still have that other strand of DNA, and so your body is able to use that other strand as a template for making the correction. And so as long as these corrections get fixed before the error gets passed on, all's okay. If uracil was in DNA, well, then you wouldn't be able to tell it that there was an error. So this is why DNA evolved to have thymine instead of uracil, because thymine, well, you've got a methyl group here. And so now when you deaminate cytosine, you get uracil, and you know that uracil doesn't belong. Because you know that you don't have uracil, you have thymine, and uracil doesn't have that methyl group there. Of course, you could have like two modifications where you get a methylation and then a deamination and get really unlucky and have something that looks like thymine. Or I guess you have thymine in that case. But by having it so that DNA doesn't have uracil, that it has thymine instead, well, then you don't have that problem. And so also, if you think about, okay, well, why doesn't RNA? Well, RNA doesn't need it because RNA is just temporary. And so why go through the hassle and cost of adding a methyl group if you just have a temporary copy? And so RNA can be a little more like, it's okay if we make a mistake and can't fix it because it is temporary. Whereas the DNA, we've really got to fix it. So our cells are really, really good at fixing it, and they have different fixing machineries depending on the type of damage. You still have that other strand of DNA, and so your body is able to use that other strand as a template for making the correction. And so as long as these corrections get fixed before the error gets passed on, all's okay. Another modification we don't really want to happen is spontaneous depurination. So we talked about how we could kind of make spontaneous changes in the bases. Well, here we're actually losing the whole base itself. Through hydrolysis, it basically just gets chopped off by water. And this is going to leave you with an apurinic or abasic site. So if you had a pyrimidine be lost, it would be an apyridinic site, but it's more common in the purinic sites. So our G or our A kind of just going bye-bye. And then your cells can recognize that. They can also recognize when damage kind of caused by UV causes there to be adducts or kind of the bases stick to one another permanently. UV light, ultraviolet light, highly, highly energetic. So remember that we can measure the concentration of nucleic acids like DNA and RNA by shining ultraviolet light on them and seeing how much they absorb. Well, it turns out that your DNA and your RNA can absorb UV light from the sun and things. And when it absorbs the light, sometimes what happens is those bases get so energetic, they get so excited that they link up to one another in covalent bonds instead of just forming those intermolecular interactions with one another. You can get things like these photoadducts or these thymine dimers. So thymine is particularly notorious about doing things like this, where it's going to form these covalent bonds with, say, other thymine residues. And that is going to cause this problem that your cells need to be able to recognize. And thankfully, it causes this weird bulge in the DNA so that your cells can recognize it. And they're like, hey, I know that that's not supposed to be there. And then they can go and fix it. And they know what to add, what the right thing is, because they have the other strand of the DNA telling it what it should be. But if they don't fix it in time, well, then you can get mutations passed on, and these mutations might be carcinogenic. So quick terminology note, something is mutagenic if it can cause mutations, but not all mutation causing things will actually cause cancer. And so things that are kind of known to cause cancer are carcinogenic, so capable of causing cancer, as opposed to just mutagenic, which is capable of causing mutations. And so sometimes studies might find that something is mutagenic, but they haven't actually yet shown that it's carcinogenic, so that those mutations are actually capable of causing cancer. if we talk about the mutations, if it changes a purine to a pyrimidine or vice versa, we call it a transition because you're transitioning to a new type. You're going from a purine to a pyrimidine. If it goes from a purine to a purine or from a pyrimidine to a pyrimidine, we call it a transversion because you're changing to a sort of new version of the same type. And this can occur through things like UV, methylating agents, and by random spontaneous events. When you have these changes, then they're going to make the DNA letter look like a different DNA letter. And so then when the cell goes to copy it, they're going to copy the wrong letter, unless your cells can fix it first. And so your cells have a lot of fixing machinery that can do that fixing. And you'll learn all about that if you take molecular biology. We've been focusing on the bases, but we can also have damage to the strands itself. We can get strand breaks. This is especially common with RNA, and it's one of the reasons why RNA is more fragile than DNA. Remember that in addition to having a U instead of a T, RNA had that right foot. It had that 2' OH, and it turns out that its right foot can kick its left foot, and it can kick the RNA, the next RNA in the chain off. This is especially common under high pH, so our basic or alkaline conditions. If we have this 2' OH be deprotonated, well, now we have a strong nucleophile that's going to attack this phosphorus, this electrophile. So we get this kind of like interchain breakage because, well, then that phosphorus is going to kick off the next base in the chain. We start off this cyclic product, but then that phosphate group can end up on either the 2' position or the 3' position. And so this is going to lead to problems if we were to rely on RNA as being a long-term storage form. DNA, however, we don't have that problem. It doesn't have that O, so it can't kick off the next letter. And so with your DNA, you don't need to worry about this base catalyzed hydrolysis. With RNA, even if it's not basic conditions, you can still have proteins that are actually going to degrade the RNA. So one of the things we worry about a lot in the lab is RNase A. It's this ubiquitous, so it's basically everywhere. It's this RNase, so it's an RNA chewer that basically what it does is it deprotonates that 2' OH and causes it to attack. And so this is an enzyme catalyzed version of what we saw before. Even if you don't have the sort of chain breakage at the altered pH, if you do have an alteration in pH, it can affect the base pairing and it can cause denaturation. Those backbone phosphates have a pKa near 1, so they're going to pretty much always be negatively charged. But those bases, remember we call them bases because those nitrogens can take protons, just not very readily. But if the pH is low enough, then these can get protonated. And if the pH is high enough, they can actually deprotonate the protons that they already have. And so either of these were going to disrupt your hydrogen bonding, leading to denaturation. I don't have time to go into the details, but there are a lot of different nucleic acid-based therapeutics, things like mRNA vaccines, where we can give cells mRNA and get them to make specific proteins. There are also things where we can give cells smaller pieces of RNA, things like microRNAs and siRNAs that can be used to silence gene expression through RNAi. There are also things like antisense oligonucleotides that can alter how different RNAs are processed. Lots of cool stuff. However, because RNA has those fragility issues, there are also modifications that are often made to nucleic acids that are used for therapeutics. These include things like changing that 2' OH position so it's less kicky, putting in like a methyl group or a methoxyethyl group, as well as changing some of the linkages to be more stable so they can't get kicked off as easily, doing things like base modifications to make them so that the body will not think that they're as foreign, and other things, and conjugating them, attaching them to various sugars and things like this to help them get into specific cell types. So to review, our nucleic acids, DNA and RNA, play a number of important roles in the body. Most classically, our DNA genes are going to serve as instructions for making proteins, with messenger RNA serving as a temporary intermediary that brings the instructions for making the protein to the ribosomes, which actually make the protein. There are other roles, however, of RNA, such as functional RNAs, such as transport RNAs and microRNAs, and ribosomal RNAs, because that ribosome is actually a complex of RNA and protein, and the RNA is actually the part that's playing the catalytic role. So RNAs can be enzymes, and so we have ribozymes, which are RNA enzymes. So not all enzymes are proteins, but most of them are, as well as biotechnological means by which RNA can be used to do things like make vaccines and treat various diseases. RNA and DNA, their different functions are well suited by their structures, or I guess their structures make them well suited for those functions. So with RNA, we have a bit more flexibility. We've got that 2' OH, which, yes, it makes us more fragile. It makes us prone to kind of breakage and RNases, but it also provides additional opportunities for binding. RNA tends to be single-stranded but fold up into complex structures, whereas DNA, it's usually double-stranded, and the double strands kind of come together in this anti-parallel fashion, form a double helix that then gets wound around proteins called histones, which gets wound up even further into tightly coiled chromosomes. You can kind of see the sequence through the grooves of the DNA helix, and so proteins called transcription factors are able to go and read it and recruit RNA polymerase to come transcribe various regions. However, in order to actually gain access to those regions, the chromatin structure needs to actually be modified. Those nucleosomes have to be moved out of the way. And so chromatin remodelers can help with this. Chromatin remodeling complexes can come and move those histones out of the way. And one of the ways where they know where to go is through modifications to the histone proteins. The tails of those histones have a bunch of lysines that can get modified. And this can alter the accessibility of regions so that those transcription factors can come read in the window. Then the DNA can get unzipped and RNA polymerase can come make a copy. And RNA polymerase can make a copy and DNA polymerase can make a copy, and you can make these copies because of the complementary base pairing relationship between DNA strands, between DNA and RNA. And this also makes it so that if you have modifications to the bases that you don't want, you have mutations, you have things like this, that then you're able to fix the DNA, hopefully before it's too late, because you have the other copy you can use as a template. However, if those mutations don't get fixed in time, well, then you can get permanent mutations. And these mutations may or not cause changes in the protein that gets made due to the degenerate nature of the genetic code, where you have multiple codons encode for a single amino acid. Our cells are very good at correcting the damage, however. And so thank your cells, your awesome little machinery inside of your cells, and thank the DNA, thank the RNA for having this awesome structure that makes it so that all this can happen.